Hello, my name is Juan M, and today I'm going to talk to you about how to leverage Aphrodite to identify bias in statistical phenotyping algorithms. This is work that's been done with Nigam, Shah, and Vyjeyanthi at Stanford, and it's been funded by National Institute of Aging. So why is this work important? Well, in the last couple of years, and more frequently in the last couple of months, we've seen that there's all this AI bias that's been found on algorithms ranging anywhere from image cropping, image selection, to job hiring and things. So if those algorithms have some sort of AI bias due to underlying data distributions, what's happening with our phenotyping algorithms? Well, let's find out, right? Here, the important part of this work is that, we're not trying to find the best performing phenotyping algorithms here. We're trying to show if any of these algorithms are fair to underrepresented minorities by, showing that, they make the predictions in the same way, but they have the same metrics for all these races, right? The data we use Stanford Healthcare's data, EHR data, 3 million patients, everything, all the medications, codes, and lab records, and the data and the CDM V5.3. Phenotyping methodology, we use probabilistic phenotyping via noisy labeling using the APHRODITE package. And the big picture here is that if you're not familiar with APHRODITE, is that instead of using a rule-based definition, we use a labeling heuristic to identify potential cases and controls. This heuristics is usually a set of codes, and we build machine learning models on those. At least for the phenotypes we pick dementia, frailty, mild cognitive impairment, Alzheimer's disease, and Parkinson's disease. Here are the demographics of the data set that we were using. Notice that we have very low representation, especially for Native Americans and Pacific Islanders, but in other demographics as well. We also have around 27% of the data that we don't know the race. So these are things that need to be addressed. Experimental setup. So we pick 5 phenotypes. We use cases and controls that are matched by age, gender, race, and length of record. We have classification algorithms, use lasso random forest SVM, and we did multiple scenarios to evaluate. So we basically did around 200 model evaluations. Accuracies that we're using is accuracy, metrics that we're using is accuracy, sensitivity, and specificity. But more importantly, this is this metric called variation, where we want to know how different their performance is from one model to the other on different across different races. So let's get to the good stuff. So we evaluated 2 different approaches. We build models for individual races and compare against them, and we balance our models and do a leave one out evaluation. The most important thing to know here is that for none of these approaches, race was used as a feature or variable. So this is completely unseen in the algorithm. The individual for the individual separation, the steps we did is that we removed all undefined patients. It's a good chunk of the data, but we can't evaluate them correctly. And we separate the phenotype cohorts by race, and we build models on each race. So if you notice here, primarily we have for at least, for dementia and Alzheimer's disease, we have 2 very clear differences, right? So in these plots, the redder the figure, the worse, meaning that there's the highest variance between the models. But the lighter the color means it's the lowest variation between the models. So at least for the Alzheimer's disease and dementia, we have that very low variance, and for the other ones, we have huge variance, especially for certain particular demographics. On different demographics, algorithms, we see that we get over almost 25% difference between the performance in terms of accuracy for this model. We see the same pattern for sensitivity and dementia and Alzheimer's disease, and the same pattern, in all the other algorithms where there's a little bit more difference. Caveats of this approach is that your analysis are as good as your underlying data and how reliable it is. There's a small racial representation leads to non-generalizable results, and that is one of the big ones. Phenotype identification complexity plays a huge role. I'll talk about that a little bit later. The other evaluation methodology we used was this balance and leave one out evaluation. Again, we remove all the undefined patients. We build models the traditional way using as much data as we can. We build models balancing the data by equal race distribution, and we build models leaving out a single race and that leave one out approach. So again, we see that, at least for the metric sensitivity for dementia, we have that the full model and the balanced model have very little variance. And the same thing for Alzheimer's disease. But if you look at frailty and mild cognitive impairment, you see that these things are increasingly red, meaning that there's more there's more, variance. Same thing for specificity, we have a similar pattern as well. Caveats again, it depends on how good your data is. And also building this balanced models is hard, particularly at institutions with not much diversity on in their EHR or available data. But that's why, further other research avenues is this could be overcome with federated learning or using leveraging large data networks. The general insights of this work is that, at least in this case, for this phenotypes that we picked, we found two very stable phenotypes, dementia and Alzheimer's. Since for both models, the top predictive features are just a handful, and they're found in most patients regardless of race. So that's what made those models very stable. The remaining three phenotypes, show higher variation due to the fact that there are no dominant features between them. Thus, the models built in each experiment are very different from each other. The big picture conclusion is, and what we were funded to do here is that, are the phenotyping algorithms fair for underrepresented minorities? Well, it depends, right? Mostly on the phenotype and the complexity of the underlying algorithm. It could have minimal effects or it can't. What do we do to improve the models? Well, you have to carefully consider this race distributions. You need to do thorough experimentation to assess if you have one of those phenotypes that this is going to go bad or good. Luckily, it's the full framework to do all this stuff in an automated way. It's going to be coming to APHRODITE by the end of the year. And obviously, there's way too much other details and analysis for the very few results that I showed you here. And that's going to come in a paper in a couple of months and a full paper of this. So thank you very much for listening to me. And you can send me an email, contact me on Twitter if you want to discuss or have any questions. Thank you very much for your time.